let's move to our next speaker. So we have a recorded talk from Dina Ramić. Dina is a PhD student at the Biotechnical faculty at the University of Ljubljana, and she's working on Campyl, quorum sensing, and biofilm control.
Good morning, everyone. My name is Dina, and today I will present you a part of my PhD thesis with the title Towards Development of Reliable Biological Methods to Detect and Quantify Auto-Induced HER2 Signals from Campylobacter jejuni in Food Samples. At the beginning, a few words about quorum sensing. Well, for many years, there was a belief that bacteria are unicellular organisms starting primarily to find nutrients and multiply. But today, we know that bacteria are able to communicate and coordinate their behavior, which enables them to form highly organized multicellular entity. That communication is known as quorum sensing. Quorum sensing is based on the ability of bacteria to synthesize low molecular weight signaling molecules known as auto-inducers. When the concentration of bacteria increases, the concentration of signals also increases and triggers the adaptive features of bacteria which are necessary for their survival in the environment. It's known that bacteria can produce different kinds of signaling molecules. For example, gram-positive bacteria produce auto-inducer peptides, while gram-negative bacteria produce acyl-homoserine lectins. For one group of signaling molecules, it's known that it's present in different bacterial species, and that group is known as auto-inducer 2 signals. It's also known that quorum sensing mechanism is important for the establishment and development of food microbial ecology, but is also involved in the food spoilage because quorum sensing mechanism is involved in biofilm formation, cell motility, conjunction, production of virulent specters such as secretion of toxins and proteases. But in this research, we will focus on quorum sensing mechanism based on auto-inducer 2 signaling molecules. Luxase enzyme is responsible for the production of auto-inducer 2 signaling molecules, and receptors which are known for now for auto-inducer 2 signaling molecules are the receptors which are present in Vibrios and the one which are present in ecteric bacteria. For Campylobacter jejuni, is known that it produces auto-inducer 2 signaling molecules, and the enzyme which is responsible for its production is luxase, but the receptor is not known, so the quorum sensing mechanism of Campylobacter jejuni is partially characterized. It's known that quorum sensing mechanism of Campylobacter jejuni affects many adaptive features such as stress response, chemotaxis, biofilm formation, virulence, pathogenicity of these bacteria. So it's very important to study quorum sensing mechanism of these bacteria in order to modulate Campylobacter's properties and to reduce the risk Campylobacter present for food safety. But we need to go back to some basics and find the most reliable and sensitive biosensor assay which will help us to detect and quantify signaling molecules from Campylobacter jejuni. So the objectives of this study was to optimize the biosensor assay and choose the most appropriate Vibrio harvest strain for the studying of Campylobacter jejuni quorum sensing mechanism. The second objective was to quantify auto-inducer 2 signals produced in Miller-Hinton broth by Campylobacter jejuni with biosensor assay. Then we wanted to validate our biosensor assay, so we detected and quantified Campylobacter's signal for the first time with an analytical method, and that was HPLC-FLD. And the fourth objective was to detect and quantify Campylobacter's signals in the food model with biosensor assay. For food model, we used 5% chicken juice in Miller-Hinton broth. Like I said, we used different Vibrio harvest strains for the biosensor assay. I will say a few words about Vibrio harvae quorum sensing mechanism. This bacteria possesses 3 different signaling molecules which affect the quorum sensing mechanism of this bacteria. First is the specific one for this bacteria, and that's cholera autoinducer signal. Then we have the acylhomoserine lectin, which is present in gram-negative bacteria. And the third one is autoinducer 2 signaling molecule, which is present in different bacterial species. Acylhomoserine lectins and autoinducer 2 signaling molecules are the responsible for bioluminescence response of Vibrio harvae strains. So when we add the spent medium of, for example, Campylobacter jejuni to Vibrio harvae strains, Vibrio harvae responds in the bioluminescence way. So we use 4 different strains of Vibrio harvae, and that was VB170, which is Luxan mutant, the mutant which not recognize acylhomoserine lectins, but is able to produce autoinducer 2 signals and is able to recognize those signals. Then we have Vibrio harvae MM30, that is Luxas mutant, the mutant which is not able to produce autoinducer 2 signals, but is able to recognize them. Third one was Vibrio harvae BB152, that is Luxan mutant, which is not able to produce acylhomoserine lectins, but is able to recognize them as well as to recognize autoinducer 2 signals. And the fourth strain was Vibrio harvae MM32, that is Luxam and Luxas mutant, which is not able to produce acylhomoserine lectins and autoinducer 2 signal molecules. So we added the spent medium of Campylobacter jejuni to our Vibrio harvae reporting strains, and we determined that Vibrio harvae MM30 has the most sensitive and reproducible response to spent medium of Campylobacter jejuni. Also, the differences between the response to the spent medium of reference strains and its Luxas mutant were the most high. So differences were more than 2 logarithms. Okay, so we choose Vibrio harvae MM30 for further experiments, and we use that biosensor assay to quantify signals in Miller-Hinton broth. So first, we use the spent medium of Campylobacter jejuni in different volumetric concentration. Then we also added to our Vibrio harvae strain standard molecule, that's DPD. DPD is a precursor of autoinducer 2 signal molecule. We added that standard to our Vibrio harvae report strain different concentration. Then we fitted our data with Hill model, and by comparison of the response to DPD and response to the spent medium of Campylobacter jejuni, we determined the micromolar concentration of autoinducer 2 signals. Then we decided to validate our quantification with biosensor assay with analytical method with HPLC FLD. First, we needed to detect those signals with HPLC, and we were successful. We detected the signal at the same retention time as for DPD, which was positive control. And we also use a negative control, that's a Luxas mutant from Campylobacter, which does not produce signal molecules. And what we could see, there is no signal in the exact retention time, like there is here for spent medium of Campylobacter jejuni and DPD. Okay, we were also able to determine micromolar concentration with HPLC FLD, and this concentration was not significantly different from the concentration determined with biosensor assay. So that's why I decided to use biosensor assay for quantification of signals in food model. The principle of quantification was the same as the biosensor assay, which I mentioned before. And we were able to detect and determine micromolar concentration of autoinducer signals in the food model. Okay, To conclude, this study for the first time highlighted the usage of biosensor assay for out-inducer two-signal quantification. This is methodological novelty in the field of cell-to-cell communication in Campylobacter jejuni, and it's crucial for further studies. The use of this biological method represents a reliable and affordable method that can be used to detect and quantify signaling molecules in food sample, because now we do not need to use expensive and more complicated methods like our, for example, analytical methods. And this biosensor method is also faster than analytical method because we don't need to prepare the sample for analysis. Okay. At the end, I would like to thank my mentor, Professor Sonia Smolemogina, my mentor, Dr. Istok Dokša, and my colleague, Dr. Anja Klančnik, who supported me. And I also want to thank to Slovenia Research Agency for financing this project. Thank also to you because you are listening to me. And if you have any further questions, please write me to the mail, which is written below.
Thanks a lot, Dina, for your talk. Are you actually here? We have some questions from you. So if you can unmute your... Perfect. So I'm just going to read the first question that I have here from Desi. Very interesting talk. How many isolates do you need for them to start acting as quorum sensing? And do you have any model that illustrates how many isolates are needed to act efficiently?
Okay, so if I understand it properly, it's about colony forming units. So what we saw is that when we have 10 on 8 colony forming units per milliliter, then the concentration of signal is high. And if we have lower than 10 on 8 colonies, then the concentration is very low. So I propose that if we have 10 on 8 bacteria, then the quorum sensing is triggered.
Thank you, Dina. We have another question from Arnold. So Vibrio can pick up Campylobacter AI2 signals, but do Campys pick up the signals themselves? It could be a way of influencing other bacteria while being deaf to the signal itself.
That's a very interesting question. I suppose that Campylobacter can also recognize its own signals. Yes, Vibrio is recognizing them because autoinducer 2 is universal signal and is present in more than 70 bacterial species. It's true that it's not known the receptor for Campylobacter, but I propose it has, because if we have Luxas mutant, then we can see that the production of biofilm is lower, the motility is lower, and also some virulence factors. So, for example, invasion to the cell is also lower if we have Luxas mutant.
Thank you, Dina.
Welcome.
You can continue and you can follow up the discussion on Discord.